Well, hello everyone. Here we are at the Federal University of Bahia in Brazil at the graduate program in food science in the course Topics in Food Science. I'm Professor Nelson Colauto, responsible for this course, and this is part of the initiative to internationalize the graduate program in food science. Today we have the pleasure of welcoming Professor Nicolas Bordenave with an important topic from Ottawa, Canada. Professor Nicolas Bordenave is Associate Professor from the Faculty of Medicine, Department of Biochemistry, Microbiology, and Immunology from University of Ontario. Professor Bordenave, be welcome, and the floor is yours.
Thank you very much. So thank you for the presentation. Sorry again for being late. As I was saying, my network was playing tricks on me, but we've made it. So I will be able to talk to you about some of the things I've been working on lately over the past few years. Trying to bridge food science and nutrition, and hopefully you will find that interesting and that will trigger some questions. But thank you very much anyway for welcoming me. And just so that you know, I'm in transition now. In a couple of weeks, I'll be leaving the University of Ottawa and moving to a new research position in France. So things are changing, research programs are changing, but what you will see, I hope, is something that will carry over to the new place, because as you will see, we'll talk about dietary fibers in polyphenols, and these don't change whatever country you are in. So hopefully that's something that will stay current. So I'll talk about delivering the functionality of dietary fibers and polyphenols, like as you can see on that slide. I'll get you a bit up to speed on these functionalities, so everyone can understand what follows. But first, I'd just like to say a couple of words that all the work that you'll see is work that's been done with students. First and foremost, a lot of students have participated to that. Collaborators also across the University of Ottawa, Purdue University in the States, the company PepsiCo as well, and just so that you know, part of that work has been funded by PepsiCo and by a company called AgriFiber Solutions. So a part of that has received private funding. So it shouldn't change anything as far as scientific results are concerned, but it's probably better that you know. So let's start the scientific part of that. So I'll talk to you a bit about the role of dietary fibers and polyphenols in satiety and control of glycemia. Satiety and control of glycemia are a part of, they are at that boundary in between food science and nutrition, because we know that satiety and glycemia are intimately related to health issues such as obesity, type 2 diabetes, chronic inflammation, and they are satiety and glycemia are very much controlled by the type of food that we eat. And so this is how the food that we eat can influence phenomena just like satiety and glycemia, and therefore influence health outcomes. And in particular, fibers and polyphenols. So what you can see on the left-hand side is the role of dietary fibers. Particularly the soluble dietary fibers. When we digest the food that contains soluble dietary fibers, they tend to build up viscosity in the stomach, in the intestine. There are multiple physical mechanisms that explain that, but essentially it's through that viscosity buildup that soluble viscous dietary fibers promote satiety. There are meta-analyses that show that about 80% of the variance of satiety and glycemic response are explained by the viscosity of soluble dietary fibers. So, on the one hand, we have that soluble dietary fibers that we want them to be solubilized and to increase viscosity in the gut. On the other side, we've got phenolic compounds, and these are known to inhibit digestive enzymes. So, for example, they come and bind the enzymes that are in the stomach and in the small intestine, and therefore they slow down the digestion of food, and therefore they promote satiety, and they promote more moderate and longer glycemic response. And they do the same with glucose transporters in the intestine as well. So what happens is that there are physical effects from the fibers through viscosity buildup and biochemical effects from the phenolic compounds that inhibit digestive enzymes and glucose transporters. And all that combined, we think, is can promote a slower digestion, slower transport of nutrients to the blood, and therefore increased satiety and better control of glycemia. And the interesting thing is that these fibers and these phenolic compounds are very often present in the same foods. They are either co-ingested or co-formulated. For example, here you've got a breakfast drink that's made of oats, so something very rich in fibers, with berries. And berries are known to be rich in polyphenols as well. So in the same food, in the same drink, you've got the fibers and the polyphenols. Or you can have oatmeal, for example, something like here, or whole wheat bread, like you can see here. And if you eat that food that's very rich in fibers and at the same time drink tea or coffee, then you add another source that is of polyphenols, and therefore you co-ingest the polyphenols and the fibers. But the point of that example, of these examples in particular, is just to show that it's very common to have both fibers and polyphenols at the same time in the stomach, in the small intestine, and both are known independently to promote satiety and promote a better control of the glycemic response. Is that so? I'll just pause here briefly. Is everything all right so far? Is that clear? Am I talking too fast? That's that's something I should check. Just let me know if there's anything.
Yeah, it's all right. But if you could slow down a little bit, it will be better.
Okay, I'll slow down a little bit then. I know, as I told you, I think that was last October or so. Yeah, 95% of my students tell me that I speak too fast. So that was a good reminder that I should slow down. So I will slow down and keep going. All right, thank you for the feedback. So as I was saying, it's very we've got fibers and polyphenols that are known to promote satiety and to promote a better control of the glycemic response independently. But what is known as well is that they can engage in molecular interactions. The polyphenols and the fibers can associate at the molecular level to form complexes. So what we'll try to see is what is the nature of these complexes and what is the impact on first on the polyphenols and then on the fibers. And what does that say of the functionality of the fibers and the polyphenols? So let's see that. First, the nature of the molecular interactions. So as I was saying, if you have a dietary fiber, which is essentially a polysaccharide, it's the that kind of blue chain that you can see here, and polyphenols like this one. This one is gallic acid. It's a phenolic acid. What we've done is that we've put them together and measured their association. It's something that's quite easy to do with a technique that is called isothermal titration calorimetry, and that enables to measure what you can see here, the parameters of binding. So we have fibers like oat beta-glucans, guar gum. We've got phenolic compounds from vanillin to a compound that is called EGCG. It's epigallocatechin gallate from green tea. They're bigger and bigger. And we tried to put them together so that they would form a complex, as you can see here, and measure by ITC, measure the characteristics of binding. And so I'll summarize that briefly, but what you can, the lessons that we've taken from that is that when polyphenols and fibers are put together, they complex automatically. That complexation is spontaneous. There's no chemical reaction. There's no need for heat. Nothing. It's just when they are together, they just bind. They just associate and form a complex. That complex is non-covalent, which means that there is no new chemical bond that is formed. It's only by hydrogen bonds or hydrophobic interactions. So there's no chemical reaction really. It's just that they associate, just like magnets. So that's how they form complexes. And that complexation is complete, which means that the equilibrium is more towards the formation of the complex. There's very little, after you put fibers and polyphenols together, they associate, and there's very little free polyphenols left after that. As I was saying, it's essentially with hydrogen bonding, with phenolic acid in particular. And I'll skip over the other details. But what we know is what we've shown with beta-glucans, guar. These are some polysaccharides. What we've shown with phenolic acids like vanillin or gallic acid, and up to flavonoids like the green tea flavonoids, we know that happens too with pectins, that happens too with tannins. So it's really a general thing that whenever fibers and phenolic compounds are together in the same environment, they just bind. And so this is what is written here. It happens all the time. Whenever they're together in the food, or whenever you eat something with fibers and eat something with polyphenols, then they meet in the stomach and in the small intestine, and they bind. So when you have fibers and polyphenols, you have that kind of complex. But the question is, the properties that we know for fibers and the properties that we know for polyphenols, are they still true when fibers and polyphenols are together as a complex? Because many times in many studies, they are studied in isolation. Researchers tend to take fibers and look at what they do. And so we know the property of fibers. They take polyphenols, look at what they do, and we know the properties of polyphenols. But when they are together, they are no longer two independent things that can do whatever they can do. They form a complex. So the question that we were asking in my lab is, do the polyphenols and the fibers have the same property related to nutrition when they are in the form of a complex? And so this is the next part. The next part being when the polyphenols and the fibers are complex, they form a complex, do the polyphenols behave the same? That's one of the big questions. So I'll just pause here again just to ask you, is that I've tried to slow down. Is that slow enough, or should I slow down again a bit more?
It's fine. It's perfect. It's a good time.
Okay, excellent. Excellent. So what we'll see is the impact of that complexation on the polyphenols, on the functionality of polyphenols. So I'll show you an example that is taken from orange. So this dates back from the time I was working in industry with PepsiCo, and you may know the brand Tropicana for orange juice. I don't know if you know that, if you know the brand. I know that Brazil is one of the biggest producers of oranges for the Tropicana brand. But most of the Brazil oranges go to make the juice in Europe. The juice in the States is mainly produced in Florida. But yes, it was related to the Tropicana business. The question was, when from the orange we make the juice, there's no more fibers. But we know that fibers are important for, as I told you, for satiety, glycemic response, and so on. So the question is, can we add back the fibers into the juice to promote satiety and a healthier beverage? So to do that, we had 3 different products. The first one was orange juice by itself, so with no fibers. The second product was orange juice with orange pomace. So the pomace is what is left after you squeeze the orange. So you squeeze the orange and you've got 2 things left: the peel and the pomace. The pomace is the fibrous material that is left and that you would normally eat. Like you can see these white segments and that kind of things. So we had orange juice with the pomace added back into the juice, and as a control, just the regular orange fruit. So in terms of composition, this is just the juice without fibers. This is the juice with the fibers from the fruit, and this is the fruit. So in terms of composition, this one, the fruit, let me just switch to the pointer. That will be easier. This one, the fruit, and this one, juice plus pomace, are equivalent. It's just the same composition. Same sugar, same fibers, same flavor, and all that. It's just that in this case, first the juice was extracted, then the pomace was recovered and added back. But in terms of composition, it's the same. The thing that differs between the fruit and this is the processing. That here the matrix is intact, whereas here it's all ground and made into and finely microground, I would say, put into very fine particles. And back to the question: what is the impact of fiber polyphenol complexes on the polyphenols functionality? We looked at one polyphenol functionality, which was the inhibition of glucose transport by the polyphenols. So maybe it's worth saying a couple of words about that. As I was saying at the beginning, polyphenols. So in the small intestine, whenever there's sugar, the sugar is taken by proteins on the walls of the intestine that transfer the sugar to the blood. So these are glucose transporters. But we know that polyphenols can come into these transporters and block them from transporting glucose from the intestine to the blood. So this is one of the properties of polyphenols, that they can block the transporters from transferring glucose from the blood, from the intestine to the blood, and therefore they slow down the appearance of glucose into the blood, which is a good thing. So we've looked at these three types of products, and first we looked at the polyphenols. So the polyphenols, if you look at the orange juice, let's not look at the orange bars, just look at the green bars with the numbers. This is the amount of polyphenols that are available in the small intestine. So from the orange juice, only 10% of the polyphenols from the orange juice are free and available in the small intestine. But as soon as you add fibers, whether it's in the fruit or in the juice plus pomace, then that fraction of available or accessible polyphenols jumps up. It's almost by a factor of 3. So when you add fibers, it helps polyphenols being available. It makes them more stable. It helps them being solubilized in the small intestine. So it's better for making polyphenols bioavailable. So very low availability in the orange juice, and as soon as you put fibers, that availability goes up by almost a factor of 3. However, if we look at glucose transport, I'll explain you this chart. What we see is the contrary. So we've used intestinal cells to measure how fast glucose goes through the cells, and to simulate how the glucose is transported from the intestine to the blood. And that black line here is the control. So this is just glucose. And you can see that this is the reference. This is the speed at which glucose goes across the intestine to the blood. And what you can see here, that dotted orange line is orange juice. So orange juice is much slower. So there's not a lot of polyphenols available, but the ones that are available can slow down glucose transport considerably. If you look at the two others, polyphenols are more available, 3 times more available, but they don't slow down glucose transport as much. When there are fibers, the transport of glucose is closer to the control. So if we look at that, no fibers, very low availability of the polyphenols, but efficient slowdown of glucose transport. And when there are fibers, higher bioavailability, but less slowdown of glucose transport. So what does that mean? It means, so I'll spare you the details, but it means that fibers just bound the phenolic compounds. They form that complex. And because they formed that complex, it looked like the polyphenols were more available. They were less degraded through digestion. They were more solubilized. So everything looked like the polyphenols were more available thanks to the fibers. But at the same time, the fibers kept them, stuck to them, so that the polyphenols could not go and block the transport of glucose. So it's the availability here was actually a false availability. It looked like the polyphenols were more available with fibers, but actually they weren't, because they were kept by the fibers. So what we see in that case is that we know that fibers are good when they build up viscosity. We know that polyphenols are good because they can slow down the transport of glucose, but when they are together, the fibers keep the polyphenols and prevent them from slowing down the transport of glucose. So it just looks as if fibers were preventing the polyphenols from doing their job, which is somehow a negative thing. So that's a first approach of saying a good thing, fibers, and a good thing, polyphenols, together give a not so good thing because they stay stuck and the polyphenols cannot work. So that's the first thing. The second one is looking at, we just look at the polyphenols, putting the polyphenols and the fibers together and look at the polyphenols. Now we do the same thing. We put the polyphenols and the fibers together and we look at the fibers. So what we see here are blends of fibers. So these fibers are fibers from oats. They are beta-glucans. And we put them in presence of some known polyphenols. What you have here is the reference, so just the fibers themselves. And with any kind of polyphenols, when you add polyphenols, viscosity decreases. And I remind you what I've said at the beginning, viscosity is key to the properties of the fibers. What we want is for fibers to hydrate, solubilize, develop viscosity, and the more viscous it is, the better for satiety. But what happens here, if you look at just fibers, as soon as you put polyphenols, viscosity decreases. So it's really, in that case, adding polyphenols to the fibers is counterproductive. That is, I'll just add one word to that. I'll skip on that pseudoplasticity. It doesn't matter much here. But adding polyphenols, not only does it decreases viscosity, it also reduces water holding capacity. The fibers have that capacity to hydrate and keep water around them, which participate to viscosity, actually. So they become thick and they keep water around them. And when we add polyphenols, they don't keep water as much around them. That is just by binding with polyphenols. So we looked at that at the molecular level, and what we found is that if you have beta-glucans, in that case, and this is just fibers, they are dispersed or solubilized in water. And when you add polyphenols, and I hope you can see that they're these small green triangles, actually they aggregate around the fibers and make so that the fibers aggregate together. And when the fibers aggregate together, they are no longer able to build up viscosity, hold on to water, and do whatever we want them to do for nutritional purposes. So again, that's something that is counterproductive. We want fibers to build up viscosity, to keep water, and when we put polyphenols with them, they just aggregate and they can no longer do that. So that's a problem. There's just another thing here about polyphenols and fibers. I'll go briefly over that. I'll make it really, really simple. You don't have to look at the charts. The main message here is that the effect, hold on, I'm trying to see here how I can make it shorter. Yes, let's go here. What you can see here, that the viscosity of the fibers decreases in presence of polyphenols. There is if the fibers are very diluted, that mechanism of aggregation doesn't work very well because the fibers are too far apart, and so the polyphenols cannot really aggregate them. When the fibers are very, very concentrated, the contrary, well, all the fibers are too close together so that the polyphenols don't work very well also. But there is a specific concentration of fibers where that effect of aggregation is maximum. And what we found is that really bad luck. That concentration where the effect is maximum is exactly, so we found that it's around 0.5, 0.6% for beta-glucans, 0.6, 0.8% for guar, 0.1, 0.2% for xanthan, and it's right in the range of how fibers are used in foods, generally between 0.1 and 1%. So it means that when we have fibers in foods, we want them to build up viscosity. When we add polyphenols, the polyphenols aggregate the fiber and prevent them from building that viscosity. And that effect is maximum for a certain concentration of fibers. Bad luck, this is exactly the most common range of concentrations that we find in foods. So once again, it's rather counterproductive to have fibers and polyphenols for the fiber functionality. One of the questions here, we saw that the viscosity of the fibers is higher than the viscosity of the same fibers that are complexed. It promotes aggregation, it reduces viscosity, it's maximum around a specific concentration that is quite common. But the thing is, does it matter? Is that something that will have an impact in foods? Because something I haven't told you is that everything that I've shown you so far is just the fibers in water with polyphenols. But that's not food. In food, there are many other things. There are proteins, starch, many other compounds that can interfere with that. So what we wanted to do was to go from that theoretical approach. Polyphenols can reduce the viscosity of fibers in solution, to something that is more in a food, just to see if that still works. So something that we did was to measure the viscosity of oatmeal, so oats, a oat porridge, measure its viscosity through digestion. So what you see here on that axis is the measurement of viscosity. So we're putting oat porridge with digestive enzymes, and we're just measuring viscosity. And this is the viscosity profile of oat porridge. And what you can see is that there is an initial drop of viscosity when starch and proteins are digested. But at some point, there's that increase in viscosity. This is due to the solubilization and release of dietary fibers. And this is exactly what I was telling you at the beginning. What we want from a food that has fibers is for the fibers to be hydrated, solubilized, and build up viscosity so that it can have that effect on satiety. And this is exactly what you can see here. When you eat oat porridge, viscosity first drops because the main components are digested, but then it goes back up again because fibers are released and build up viscosity. So this is just porridge, and what I will show you next is the same thing measured but with polyphenols. And with all these lines you can see here are the same porridge but with polyphenols. Vanillin, ferulic acid, gallic acid, and different kind of polyphenols. And what you can see is that in the end, the black line is still the viscosity of just the porridge, so the fibers of the porridge. And in presence of polyphenols, the viscosity decreases as well. And we can lose up to 38% in the final viscosity that final viscosity is linked to glycemic response and satiety. So it means that just the presence of polyphenols, even in food, like what I've shown you before, the presence of polyphenols in complex food still works at aggregating the fibers, decreasing their viscosity, and therefore, maybe, we don't know yet, but maybe it will decrease the satiety effect. So if you eat porridge, oat porridge, or if you eat the same porridge but with coffee or tea, will the polyphenols of coffee and tea decrease that intestinal viscosity and just cancel the effects on satiety? And so this is still a open question, and we're trying to answer it with a clinical trial right now, and I will have answers soon, hopefully. Now, I would like to finish up with a positive thing, because so far, what I've told you is that when we have fibers and polyphenols, they form complexes. And with respect to satiety and control of glycemia, well, that complex reduced the functionality of the fibers by reducing the viscosity, but also reduced the functionality of polyphenols by keeping them and not making them available to go and inhibit enzymes or digestive enzymes. So apparently, it's all negative. The problem is, as I told you at the beginning, it's so common to have fibers and polyphenols in the same food or in the same meal that it looks like, anyway, having them together is counterproductive. But we know that epidemiological data say yes, it's better if you eat more fibers. It's better if you eat more polyphenols. So epidemiology says the contrary. The lab says the lab and the test tube say when you put them together, they sort of cancel each other. But epidemiology say, well, the more you eat them, the better you are. So there's something contradictory here. Something that says, well, it looks like they don't work well together, but at the same time, there's proof that they actually do work well together. So the big question would be, what's happening with them? How do they work? And one of the hypotheses that we're trying to test now is that the complex between fibers and polyphenols actually a carrier of the polyphenols and the fibers towards the lower gut microbiota? I'm sure you've all heard of the gut microbiota, the billions and billions of microbes that we have in the large intestine that are intimately related to our health. The hypothesis that we have now is that, well, fibers and polyphenols kind of protect each other when they form a complex, and they may not work as we thought they would in the upper gut, the small intestine, but they may just protect each other to work with the gut microbiota, because then the gut microbiota can use the polysaccharides and ferment them, and can use the polyphenols that come with them. So is that complexation between fibers and polyphenols actually a good thing? Not where we thought it could be, in the upper gut, but in the lower gut with the gut microbiota. So that's the kind of things that we're trying to test now. But it's still, there's still open questions. For example, what you can see here, I hope that you can see that there's that heart logo here on that porridge box, because fibers are you can have a health claim with fibers. Fibers are good. A good amount of fibers, of soluble fibers, can reduce cholesterol. But when this is tested, it's tested. The clinical trials are made. When people come in for a clinical trial, we ask them to eat, for example, oat porridge, and we give them a glass of water. This is fine. We test the fibers, but this is not real life. In real life, they would eat the porridge and eat either tea or coffee with it. Would we still see the effects? So the health claims that are associated with that, like that heart logo here, is that still valid if we test the food as it is consumed in real life? That is not sure. And maybe the foods should be tested differently from now on. I've got another side that's probably more about product development. I'm not sure. This is, I'll go through it quickly so that at least maybe that's something that we can discuss. So that fiber and polyphenol story can have an impact on how products are made. So from a nutrition perspective, it will probably help better understand the soluble fibers and maybe maximize the effects of the fibers. For example, you can choose the fibers that interact the least with the polyphenols that you have in your product. So you can still have some viscosity and still have a claim. So knowing better these fiber polyphenols interactions can be important to maintain good nutrition. And on the other hand, it can be important for product development, because if you put fibers and polyphenols together, very often the fibers are used to adjust the texture and the mouthfeel so that consumers like it. But if they lose that property when they are mixed with polyphenols, you lose it. So maybe knowing a bit better about that polyphenol fiber complexation could help come up with better formulation and maybe cost saving for product development. So in conclusion, I would say that we've got strange results showing that fibers and polyphenols together in the lab actually sort of cancel each other, although we know that they're still beneficial. So that raises new questions about the fact that maybe they don't work as we thought they were, and therefore it's really important now to understand how they really work. And these are open questions. So this is from the health perspective. And from a food product development perspective, it can bring new opportunities to play with this complexation, depending on whether you want it or not. But understanding more about them will create certainly opportunities. And with that, I hope that was slow enough so that everybody could follow, and at the same time quick enough so that we don't take too much time. Is that all right?
It was great. Thank you very much, Professor Nicolas. It was very interesting to know these facts about it because, I have to change my mind about mixing everything, maybe in the breakfast at least. Very nice.
Or maybe not.
Yes. Well, now we will go to the next part of our class, and I pass to the questions of the students. We have one question from Victoria, but before Victoria, some students can write the question at the same time they speak. Then you can click on the second line that's bate-papo público. It's 2 squares. If you click that, you'll be able to see something. I'll say hello here. First, can you see my hello?
Yes, I can see it. Yeah,
Okay.
so I can follow any question that would be asked here.
Yeah, they will
No problem.
ask the questions and write at the same time, some of them. Victoria, your question, please.
Hello, Professor Bordenave. I'm Victoria, and actually I have a curiosity. During your presentation, you mentioned the polyphenols reduce the viscosity of fibers, and some of graphics shows, depending of the polyphenols, that the loss of the viscosity can be greater. So do you know why this happens?
Yes, so thank you for the question. I believe that I still share my slides, right? Yes, I can see that. So I'll go back to that, and I'll go a little bit more into the details here. So what you can see is here, the polyphenols are ranked from the smallest to the biggest here, except for this one. But what you can see is that generally, the bigger the polyphenol, the greater the loss of viscosity. And this is associated with the fact that bigger polyphenols make more complexes with the fibers. So it's a double thing. The more you form the complex, the more you lose the viscosity. And how do you form more of the complex? Generally with bigger polyphenols. Why that? Because these bigger polyphenols tend to have more, so we're going into the chemistry here. They tend to have more hydroxyl group outside, so they can form more hydrogen bonds with the fibers and form more complexes. So this is the general answer, but there are some variations depending on the solubility, for example, depending on the chemical stability. So there are variations, but the main answer would be that the more polyphenols have hydroxyl groups, the more they can form complexes with the fibers, and the more they aggregate the fibers, and the more there's a loss of viscosity. Does that make sense?
Yes, it may. Thank you.
You're welcome.
All right. Then next question is Johnny Elton. Johnny, your question, please.
Hi, Professor Bordenave. My name is John, and my question is on the chat. You mentioned that the formation of the complex between dietary fibers and polyphenols is spontaneous. Is there any process that can reduce the complexation, such as pH, salinity, etc.?
Yes, you're absolutely right. So let me go back to that slide, and hopefully we'll have something. Yes, it's spontaneous, and you can see that this is actually probably the main answer to your question. The main mechanism of binding is with hydrogen bonding. So anything that would disturb hydrogen bonding would limit that association. So in the lab, we would use urea that you wouldn't find in any food, or I hope you don't find that in any food because it wouldn't be good to have it. But any form, any compound that would interfere in that hydrogen bonding would limit the formation of the complexes, and therefore would limit that loss of viscosity or that kind of things. So pH can be one. Salinity indeed can be one because there can be interference, and other compounds. I'm trying to think here. For example, there are some. Have you heard of that? Oh, sorry, I'm missing the word in English, but EDTA, that's present in many foods because EDTA can sequester other molecules. Well, EDTA could perfectly interfere with that by taking up for example, the polyphenols or complexing them, and therefore limiting the complexation with fibers. So yes, that could happen. The only reservation I would have would probably be around pH, because as soon as you go at pH, let's say 7 and above, polyphenols are not stable. They tend to degrade at higher pH. So yes, you would limit the complexation, but not because you limit hydrogen bonding, just because you destroy the polyphenols. So it's a bit of a mixed thing. But yes, it's a very interesting approach, because that your question can suggest rightly that with the good formulation, the good mix of ingredients, then you can limit the formation of complexes and keep the activity of both compounds. So that's that could be a very good approach. Does that all make sense?
Yes, thank you. Thank you.
Thank you.
Right. Thank you. Next question is from Orlando Villela. Orlando, your question, please. I cannot see Orlando. Maybe here.
Hi, hello. Good afternoon, Professor. I really enjoyed your lecture. It's a topic that I like lots. Professor, eu vou colocar minha pergunta no chat. My question in the chat.
I have it now. Thank you. So that's about diabetic people. So let me try to say your question maybe just slightly differently. If we give polyphenols and fibers to diabetic people, is there a risk that it doesn't help? We think that it would help, but maybe there is a risk that it doesn't because of what I've shown you. Is that what is behind your question?
Yes.
Yes. Well, possibly. And this is why at the end I've shown you that. And let me go back to that slide. Here. What are the clinical results? What can we say about that? And so we have ongoing clinical trials about that, just precisely to answer your question. Because if polyphenols and fibers together don't work as expected, then maybe it's something that could be counterproductive for diabetic people. So we need to know, what we saw in the lab, for example, that kind of things. What we saw in the lab, that decrease in viscosity. How does that translate in terms of human trials, human results? And we are in a position where it is difficult, just by these results, it is difficult to answer, because a 34% loss of viscosity is quite a big loss. But at the same time, what we know is that the effects of fibers and fibers viscosity on satiety or glycemic response, these effects are visible, when viscosity is divided by 10 or multiplied by 10. It's more of a 10 factor. So this is, a 3 factor. Are we there? And we don't know. It looks like we're just at the limit. The lab results say, maybe it matters, maybe it doesn't. And we don't know yet. So we have to wait for the clinical trials. But when we have the clinical trials, the results from these trials, then we know, shall we avoid polyphenols and fibers together, or can we go with them together without any issue? Particularly for people who are diabetic and who count on that, because fibers can help them, polyphenols can help them, but we don't know about all of them together. So the question, my answer to your question is, I don't know, but we're working on it. Is that all right?
Yes, thank you very much.
You're
Okay.
welcome. And so I may just follow up quickly on that. I think this is something very important, that, very often we read scientific news about foods or food compounds, maybe dangerous, maybe not. A lot of these results are from the lab, are in vitro or in test tubes, and they don't necessarily translate in humans. There's nothing that, there's no result that is valid until it is tested in humans. You can say whatever you want about one compound, maybe super toxic or super safe or whatever, but it's only the test tube. And the gold standard is still testing in humans so that you know for real what it is. And there's generally a big difference between the test tube and the human. So I think this is, for the food scientist that you are, it's something very important to keep in mind.
Okay, you lecture, it's very interesting. Thank you.
Thank you.
Thank you. Just to let you know, because the internet was just cutting a little bit from Orlando. Orlando is a nutritionist student, and he told at first that he just loved your lecture about it, and just very, very interesting. Now we have just jumped Coach V, and Edna has some problems with internet and put the question, write the question at the left side. Could you read it?
Yes, I have it. So, how do fibers assist in reducing glucose in the gut? So, there are two things. I will try with the most complex scenario, but it can help understand the simpler. The main source of glucose in the diet is starch. When you eat pasta, potatoes, bread, whatever, cereals, starch is is broken down by the enzyme alpha amylase in small bits, and these bits are then broken down into glucose, which is transported through the intestine to the blood. And so when you eat starch that is digested very quickly, then the starch, which is a polymer of glucose, breaks down into glucose and is transported to the blood very quickly. So it gives a spike of glucose in your blood. Glucose appears very quickly in the blood. Your body, as a reaction, sends in a lot of insulin to clear that glucose. And if you repeat that too much, well, this is where the body, I'm making it simpler, but the body gets tired of sending out insulin all the time, and the beta cells of the pancreas that produce insulin, essentially don't work as well, and this is type 2 diabetes. So this is one of the main causes of type 2 diabetes. It's when glucose goes too fast in the blood. Where fibers work here, especially the viscous dietary fibers, they make a very thick contents of the stomach and the intestine. So it's harder for the enzymes to come to the starch and hydrolyze it. It's slower. But when they do it, then it's harder and slower for the glucose to come out and diffuse and get to the intestine walls and get to the blood. So it just makes everything slower. And because everything is slower, then it makes the glucose appear more slowly in the blood. It doesn't ask your body to send as much insulin. So it's instead of having a spike of glucose in the blood, it's more progressive, and it's better, generally speaking. And so this is why a higher consumption of fibers have been associated with less type 2 diabetes. So and I explained more than the fibers, but this is essentially how the fibers work with the soluble viscous fibers. They build up viscosity that makes the digestion process and the transport process slower, and therefore better for the body. Does that make sense?
Yes, thank you. I didn't have some trouble with internet, so this answer from to her. And we have the last question of Kodjvi Ayena. I don't know if Kodjvi will write the questions or if Kodjvi will talk. Kodjvi, could you give some sign if you are okay? I see you wrote. Are you able to speak? I believe he's not. Then Professor Nicolas Kodjvi just wrote his question.
Yeah, so are there any specific recommendations or guidelines regarding the combination of dietary fibers and polyphenol-rich foods for optimal health outcomes? I think this is probably the perfect ending for the last question about that, because it just resonates with what I was saying in the presentation. It was here. I'll always have a hard time saying it. Epidemiological data says that the more fibers you eat, the more polyphenols you eat, the better the health outcomes. So the thing is, with what I've shown you in that presentation, it just tells us we don't really know how they work. Maybe they don't work as we thought they were, but the evidence is still that the more you eat of all of them, the better you are. This is epidemiological evidence that is now widely accepted. And so, in terms of recommendation, this is one of the main recommendations of the World Health Organization. Increase the amount of fruits, vegetables, and legumes and whole grains in your diet. And what does it do? Fibers and polyphenols. So I think it's all consistent. The data says the more fibers, the more polyphenols, the better. And so it's in line with the World Health Organization recommendations. I don't think there's too much concern about that. What I've shown you is more of a scientific interest in terms of, do they really work as we thought they were? But it doesn't challenge that thing about eat more veggies, eat more fruits, whole grains, legumes, and everything, and it will be good for your health. That stays because the evidence is very, very strong about that. Does that make sense?
Yes, a lot. Well,
it looks like it.
With that, we finish our time, and we'd like to thank you, Professor Nicolas Bordenave, for taking time and share this lecture with us. And Professor, would you like to say the last few words?
Oh, so again, I'm coming back to that. Really sorry about the beginning and and my VPN that was playing tricks on me. But apart from that, I was very glad to share that with you. Hear your questions that were very on point, both from a food science and nutrition perspectives. I'm really glad to see that it resonates with pretty much all of you. These were great questions, perfectly on point again. So that was great to hear that. And with that, well, I just wish you all the best with your program, because it looks like you got great success ahead of you.
Right. Thank you very much again for taking time for sharing your knowledge with us, and I hope to see you next semester, maybe from another university, maybe.
All right. I hope so. Good luck, everyone, and thank you again for hosting me. Thank you.
Thank you. Bye-bye, everyone.
Goodbye.
Bye-bye.